And we move on to the final speaker of the session. One of my favorite topics. Emma Teeling talking about bats.
Thank you. First of all, I really want to say thank you to our organizers. I think we should all give them a round of applause. What meeting do you have an open bar, cocktail bar, till 2 in the morning? They've been so generous and everything has worked smoothly and it's been really exciting. But the second reason I want to thank you is you've given me a platform to tell you the story of my grand quest, my adventures into understanding the unique mechanisms that bats have evolved. Some say it's a quest, others say it's a madness, but I'm allowed to tell you about it today. And last year, I introduced the topic and the concept that bats are nature's natural model of extended health span, and that studying bats is our way to answer 2 of the greatest challenges. And what I want to do today is remind us of our challenges. Now, we all know the problem right now and why we're here is that people are living, around the world, are living much longer than before. We have wonderful extended lifespans, but our health spans have not yet evolved to meet our lifespans. So therefore, we need to find new ways to allow us slow down aging and increase our lifespans. Bats have naturally evolved this. But the second one is infectious diseases. I don't want to be a downer, but let's remember this pandemic that we've just gone through. You know, at over 6 to 20 million deaths in under 24 months because of a new novel virus that was introduced into the human species. I don't want to frighten you all, but it's estimated that there are 1 billion spillover attempts of viruses going from a non-human into a human every single year. So we need to find ways to allow us fight infectious diseases. And it doesn't matter if your chronological age is 50 and your biological age is 20, if you get an infectious disease that you can't resolve, you're going to die. That's the horrible part. But we're lucky because nature, she has found solutions, and they lie in the most wonderful order of mammals, and these are the bats. So 1 in 5 of every single living mammal is a bat. But to remind ourselves, bats live the longest given their body size of any mammal. They have the small size, really high metabolic rate, but they can live up to 10 times longer than expected. And if you're to correct for body size, the bat that holds that record is Myotis brantii. It can live the equivalent of 250 to 280 human years without showing any signs of aging. So that would be 42 years, but this species weighs 1/3 of a lab mouse. So the majority of bats have evolved ways to slow down expected aging. But also, they're resistant to many different viruses and pathogens that are really lethal to humans. I have Ebola, Hendra, SARS, Marburg. I've taken a lot of bat cells, injected bats with these infections, and they don't seem to die from it. So again, they've evolved unique ways to modulate their immune response to be able to live with these pathogens. And potentially both of these unique adaptations are linked. As we've heard, the immune response is a major driver of all the hallmarks of aging. So bats potentially have modulated their immune system to allow them to have longer, healthier lives and deal with pathogens. So I've spent really the past 20 years trying to develop bats as these new models for extended health span and also for disease tolerance. And I'm going to talk you through the things that we had to do in order to make a new model system. I'm going to tell you where we're at now moving forward and update you on some of our new results. And the majority of this is unpublished, so it might be a bit, I suppose, unrefined in parts, but you'll get the message. So how do you set up a new model system? So if you remember from last year, for the past decade, the past 11 years, myself, with this brilliant wildlife French vet, Fred Touzan, have set up a marker capture project of these Myotis bats in France. These are the longest lived genera of bats. And what we do is every year, we go to these beautiful old roosts, these colonies in these churches in France. We catch the entire population. What we're able to do is we're able to age the bats because we catch them as babies. The only way you can age a bat is shine a flashlight under its fingers. If they're not fused, that bat is under 1. So what that means, we catch the bats as babies, put in microchips, and we recatch them every year, so we know the age of all our bats. You can't age a bat any other way. We take non-lethal samples, less than 140 microliters of blood. We take wing punches. We take microbiome swabs from their mouth, from their anus. And we take a whole bunch of metadata. We feed them and release them, and we do this every single year. We now have over 6,000 samples, and we have this wonderful longitudinal marker capture project. But that's just one species. And this is one species, these long-lived bats. If you saw from the initial graph, Steve Ousted's paper, bats have different maximum lifespans, different LQs. So what we've now gone and done is extended this to have more than one study site. So we're looking at these myotis bats. These are the bats that we catch in France. They have the highest LQ. We brought in these horseshoe bats, Rhinolophus firmaquinum. This was based on this long-term marker capture project. It was a project that's been run for 60 years by a single individual, catching the bats, marking the bats, taking samples. He's very funny, and I'll tell you all about him later, maybe in a bar. We were able to go and catch Rosetta egyptianus here in Copenhagen Zoo. And of course, we started a new study of Molossus molossus. This is the shortest lived bat given body size. It's estimated to live now a maximum of 5 years. And so we were able to start these projects. What does it look like to come work in my lab? We have positions available, by the way. So we had to do an awful lot of field work from all around the world to do this. And if you see this young man here in blue, this is Ty Glonigan. He's leading these projects at the moment. And so we have to go catch the bats in France, in Panama, in the UK. We go to Copenhagen and to very exotic places. But again, we are working within these wild populations. And people say, well, why do you have to work with the wild populations? Why don't you have captive bats? You cannot keep these long-lived insectivorous bats in captivity. They don't make babies. We don't have decent husbandry. So we have to move to the wild. But you also need genomes. So I'm one of the founding directors of BAT1K. It's a genome sequencing consortium whereby we want to sequence the genome of every single living bat species to reference quality genomes. We're moving the technology forward. Thankfully, we've got a bunch of funding and a bunch of investors. And right now, we have generated reference quality genomes for a representative of every single bat species. We've got over 100 genomes that we're analyzing to uncover their unique adaptations. So we have the samples, we have the genomes. But you have to do lab validation and functional assays if you want to move forward. How do you do this? So also, let's think about it. Can you really make a knock-in bat mouse? So we've been working on this with a whole bunch of wonderful collaborators. We came up with new ways to be able to make bat iPSCs from 3 millimeter wing punches, non-lethal samples. We can reprogram these cells. We had to have a modified Yami acaperberine because the bats are different. This work was done with our collaborators Thomas Zawaka and Marion Dajouget in Mount Sinai in New York. But we're now able to take a wing punch, reprogram the cells, and differentiate into any cell type you want. So we can bring the wild into the lab. So what have we found? Let's tell our stories. So let's remind everybody about telomeres. So telomeres, remember, they're TTA, GGG. These are repeats at the end of our chromosomes. They get shorter. We all know what happens. But what do we find about bats? So our study on this long-lived population, bats we've been catching for 6-7 years, we used this relative qPCR method. And what we found is that in the longest-lived genera bats, their telomeres did not shorten with age. We couldn't make them short. We found when we looked at our blood transcriptome, because we take 140 microliters of blood, sequence the entire blood transcriptome, we found that we didn't find any telomerase expression. In our bat fibroblasts, when we looked at RNA-seq transcriptome data, we didn't find any telomerase expression. So what was happening? What was going on? We looked at genomic and transcriptomic analysis, and we implied that this enhanced DNA repair was maybe underlying this unique longevity we found in myotis. So what happened as we continued the study, as we started adding older and older and older bats? So what we found in myotis, this is about 500 samples, still the telomeres do not shorten with age in these species. So they're somehow finding a way to maintain their telomeres. What about in Rhinolophus firmaquinum? There's over 2,000 samples here. and these are some of the oldest bats because that's 60-year-long microcapture project, you find telomeres do shorten with age. So this is a long-lived species, but the telomeres are shortening with age, but at a slower rate. Rosettas, again going down the LQ, what you find is that there's a different parameter, there's a different shape to how these telomeres are eventually shortening with age. But these are captive bats, so we have to keep that in mind. And then molossus, the one I'm very proud of, because this is this new microcapture project. What we do find is you see the majority of bats only live till they're about 1, but we found some 5-year-olds kind of wrecking all our hypotheses. But the telomeres do seem to shorten with age in the shorter-lived bats. So to summarize, what you can see here is that in myotis, back here, telomeres, there's no correlation, no shortening with age. Rhinolophus formiquinum, getting a bit shorter. Rosettas eventually gets short. Molossus also gets short. So it nearly looks like the maintenance of telomeres seems to correlate with LQ. But what's driving this? So again, we look to our genomes. And what we've done is we've sequenced now 27 of our genomes are from young, so long-lived and short-lived sister taxa from multiple different branches of the tree. And we asked the question, if you looked at genes involved in maintaining telomeres, would you find that in all of the shorter-lived bats, there was some difference? What we found was quite unique. We found ORTEL1, because a lot of these protein coding genes, they really don't change. They're conserved, they're required. But we found protein level differences in ORTEL1 in all of the short-lived bats that was different than the long-lived bats. So this is something that we need to look at. So again, it's a phylogenetic independent contrast, which will allow us get access to what is the most important gene that maybe affects their telomeres. Again, ORTEL1 functions of disease, and mutations involved in this are associated with diseases of the old age. So it's important. But can we see this within our transcriptome? And what we have here, the transcriptome of myotis, this is the long-lived bat. Transcriptome of molossus, the shorter-lived bats. And these are the telomerase and the sheltering genes. And what you can see is there's a difference. There's a difference in expression. So we want to understand what's going on. Are telomeres, maintenance of telomeres required for the longest health span in bats? But I have to show this slide. I have to say it, as an omics person, RNA may not reveal the full picture. And so working with Vera and with Fatima, a postdoc in Vira's lab, what they were able to do is, again, we have fibroblasts. So you take a wing punch, grow out fibroblasts, do a trap assay on these, so not a transcriptome. And what we found was that the trap assay showed that there was telomerase activity, that you could pick it up doing these assays, but you couldn't see it in the transcriptome because of its such low level of expression. And what's interesting is you see again in myotis, the longer-lived bat, there seems to be more of it. But let's ask ourselves the question, how are bats dealing with this expression of telomerase with the lowest ever recorded level of cancer? They don't seem to get cancer. Have they evolved new ways to be able to scavenge? How can we have telomerase expression and no cancer, and when is it expressed? So again, we went and we looked at this these were horseshoe bats, long-lived bats. We were able to take samples from individuals before and after hibernation and during hibernation. And it looked like actually their telomeres were lengthening during hibernation within individuals. That means that hibernation may be some form of rejuvenation. And Sebastian Riquier, a postdoc in my lab, reanalyzed data from, it was published by a Chinese research group where they took wing punches, sequenced the transcriptome during hibernation, and again, could find an expression of telomerase. So potentially bats are using this transient expression of telomerase during hibernation to be able to elongate their telomeres. So we have genomic data. We have population-level field data, but we have to bring it back into the lab. And so we developed and optimized methodologies, again, to take these non-lethal samples and grow fibroblasts in the lab. And this is work done by Orla Alcock, who's sitting over there in my group. So let's remind ourselves, we wanted to be able to take these cells and stress them and ask the question, do long-lived versus short-lived bats show better resistance to stress? So what we would do, this is what we would expected. In blue, you would have the shortest lived bat, so it should not be that resistant. Yellow is the middle longevity bat, and red is a longer lived bat. Again, what we would do, we'd run an MTT assay, which basically looked at the viability of the cells as you add more and more and more stressors. So what did we find? So when we did this first, we used cadmium, it's a DNA stressor. Again, they're better able to repair their DNA potentially. What we found that there was a significant difference. Bats were much better able to handle this stress than were mice. And then, of course, we add in the short-lived bat. And again, we'd assumed that she would be much worse. But no, nature does what it wants. And so we had a slightly different result. So again, we need to uncover this a bit more. We did the same with tunicamycin. This is a proteasome stressor. We had these bat cells from different species to see would they respond differently and how did they respond to mice. What we actually found is that the mice seem to be better able to deal with this. Again, we add the shorter-lived bat in here, and the results, we're trying to understand what this actually means. So our preliminary results, our bats are better able to deal with DNA stress, mice are better able to deal with the proteasome stress, but Vera's group have already shown that naked mole rats actually are less resistant to tunicamycin than other rodents, and potentially because they've evolved novel ways of removing this damage. But what about mitochondria? Bats have the highest metabolic rate of all mammals. What's going on? And this is because its flight is so very metabolically costly. So work done by Sebastian and Ilse in my lab. We have 169 of our blood samples from the longest lived myotis bat. We sequenced the entire transcriptome, and we looked for the changes that were happening in genes that are mitogenes, nuclear encoding genes that are involved with mitochondrial processes and mitochondrial processes. And what we found, we looked at the top 10 mitogenes. So these genes, as the longest lived bats age, are downregulated. We looked at these pathways that are associated with age. Pathways, so the aerobic energy metabolism seems to decrease in these long-lived bats as they age. Body temperature is involved, cancer, ROS, liver disease. So you could see this signature. Top 10 genes upregulated, involved in ATP pathways, cellular scavengers, cellular signaling, and muscle fibers are quite interesting. These are upregulated as bats age. When we look at the pathways, autophagy is one that really hops out. And we'd shown before in our lab that as bats age, their ability to perform autophagic-like processes increases. Neurometabolism, cancer, hypoxia, these are all the hits that was done. This was, again, these were the hits that we found when we just looked at the overall expression changes. So there was no real enrichment. But this is kind of interesting. You find these pathways that go up and go down associated with these major different diseases. And it nearly seems that bats have evolved ways to be able to regulate and fine-tune these mitochondrial pathways that are all involved in these diseases. And again, studying bats may give us better insights. But we have to validate this. What about these bat mice? Can we actually do it? Well, a brilliant collaborator of ours, Linfa Wang, has been doing a lot of work in immunology and virology in bats. And Linfa's group, looking at all our bat genomes, found this gene, ASK2 in bats, a bat ASK2, suppresses inflammasome activity. And they made a bat mouse. They took bat ASK2, knocked it into a mouse, and then subjected these mice to lots and lots of infection tests. What you see here is that the bat mice perform so much, or they live way longer on infection. But the other very tantalizing thing is that it looks like their health span is also better. So we're looking at this. So we can now knock in some of our findings into mice and test it. But we also need to find other lab validation. How are we going to do this? And if you guys remember, Bjorn's brilliant talk, his new paper just published in Nature, where they found this dream complex that functions as a master regulator of DNA repair. Bjorn found this. Previous paper from my lab showed that when you look at the transcriptome changes with age, the one thing that was very different in bats from other mammals is they upregulate their DNA repair genes. So we asked the question, could this be due to dream regulation? Is there a correlation between bats and C. elegans? And Arturo analyzed this data. I'll just talk you through briefly. what we found is potentially yes. So what you see here, where you see the bat, the Neanderthal-human and a mouse, as you can see, these are genes that are upregulated with age in bats that correlate with genes that are enriched when you suppress dream activation in human cells. And it's brilliant. It looks like it's nearly the same thing. So this means that, ask the question, are bats repressing dream? Can we use C. elegans to understand this more? So I hope I've convinced you that the quest, the madness, is right. That studying bats will give us novel insight that allows us to understand aging better and make drugs. And I want to just show you this picture here. So I want to say that I'm not making it up. This beauty here, she is at least, so she's 14 plus years of age. She was caught as an adult, lactating, had a baby. We've caught her every single year. We take samples from her wing, we take blood, and she's completely healthy. There's little to no signs of aging. She could be 35, she could be 15. And as you can see, the one thing that the bats do, when you're going to see her now flying away, they're able to fly. So this flight potentially has driven this adaptation of their innate immune response, their ability to repair their damage, and the ability to remove their damage. And I hope I've told you that studying bats in this way will help us find our solutions. We couldn't do this without the brilliant, brilliant Bat Lab and all the people I have the good fortune to work with, and also amazing collaborators who are here. So thank you.
Thank you so much, Emma. We have time maybe for one question before the break, a very quick one.
Oh, brilliant. Oh, a very exciting idea about the dream complex. You know, I'm really looking forward to that. I wonder if you looked in your iPS cells, what happens? Is it even higher there?
I haven't looked. I haven't done that yet. That's a good question.
All right, thank you so much. I think we have to stop because we are running into the break. Thank you so much, Emma. That was an amazing talk. Let's give her another round of applause.